The overall goal of this procedure is to accelerate the detection of physiological substrates for potential lipases and phospholipases.
This method can help answer key questions in the enzymology field, such as how phospholipases contribute to lipid remodeling in biological membranes. The main advantage of this technique is that the optimization of enzymatic activity and the search for the natural substrate for the enzyme can be performed independently.
Though this method can provide insight into substrate specificity of bacterial lipases and phospholipases, it can also be modified to study hydrolysis of any other model organisms.
We first had the idea for this method when we searched for an enzyme activity that could degrade the intrinsic membrane lipid phosphatidylcholine in Synorhizobium melillotae.
To prepare cell-free protein extracts, defrost bacterial cell suspensions and store them on ice. Then, pass the cell suspensions 3 times through a cold pressure cell at 20,000 pounds per square inch. Remove the intact cells and cell debris by centrifugation at 5,000 times G for 30 minutes at 4 degrees Celsius. After centrifugation, prepare aliquots of 100 and 500 microliters from the supernatant for subsequent analysis. Set up the previously optimized enzyme assay using the pipetting schemes tabulated in the text protocol. For an initial coverage of distinct enzyme activities, use artificial substrates that yield a colored product upon hydrolysis, such as para-nitrophenol or PNP derivative.